Dr. Fatima Usha Sharif, Assistant Professor from the Department of Bioinformatics, College, Chennai. Today's topic is about the glycolysis by embedded Meyerhoff pathway. There are different phases involved in glycolysis. The glycolysis is the pathway which is involved in the split of your glucose molecule into pyruvate molecules. And this glycolysis mostly occurs in the cytoplasm of the cell, and it involves the two phases in formation of your ATPs. The first phase is mentioned as an investment phase or the payoff phase, and the glycolysis can occur both in your aerobic as well as your anaerobic state. Let us see one by one in this video today. We are going to see about the aerobic glycolysis in humans and animals, and there are different enzymes are involved in the glycolysis. The enzyme phosphofructokinase is the most essential enzyme for the regulation, and it controls the speed of the glycolysis. The glycolysis is the glycolysis which is occurred in the cytoplasm, in which your glucose is converted into two molecules of a pyruvate. One molecule of glucose can give rise to two molecules of your pyruvate. Which now let us see about the cellular level. The cellular level during the aerobic condition, the pyruvate which is derived from the glucose will enter into the mitochondria and will further undergo oxidative phosphorylation. Whereas if you take an anaerobic condition, it will result in the pyruvate which will stay in the cytoplasm and it will be converted into lactate by the enzyme known as lactate dehydrogenase. This is the schematic representation in which you can see the glucose is converted into two molecules of a pyruvate with 10 successive reactions. 10 steps are involved in the glycolysis reaction. In the aerobic condition, means in presence of your oxygen. In presence of your oxygen, your pyruvate will be converted into your acetyl-CoA. Whereas in the anaerobic conditions, there are two chances. One is the formation of your lactate, which will occur in the anaerobic glycolysis in contracting muscles and the lactic acid formations. And on the other side, you can see in the anaerobic condition, the same pyruvate will enter the formation of the two molecules of ethanol and the two molecules of your carbon dioxide, which is an alcoholic fermentation. The acetyl-CoA which is formed from the pyruvate in presence of your anaerobic condition will enter into a citric acid cycle with the formation of your 4 carbon dioxide and 4 molecules of water. Apart from that, there will be a formation of your ATPs, the source of energy to the body. The glycolysis is nothing but the glucose. You can see the various enzymes which are involved in the glycolysis. Let us see one by one. It is nothing but a metabolic pathway. By the name itself, you can clearly see it's a glycolysis. Lysis is nothing but your breakdown of your glucose from the glucose to the pyruvate molecules. As you are aware that the metabolism is divided into two types, anabolism and catabolism. Anabolism is nothing but synthesis and catabolism is nothing but degradation. The word lysis itself is mean that something is breaking down, so the breaking down into your pyruvate. Now, the enzymes which are involved, as I told you, there are 10 different consecutive steps involved in the glycolysis. The first phase is nothing but your preparative phase. The second phase is your payoff phase. The first 5 steps of the glycolysis is known as preparative phase. But energy is consumed during the conversion of your 1,6-carbon glucose molecule into 2-carbon glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The various enzymes involved are kinases, isomerases, lyases and isomerase. You can see in the second half of your glycolysis is nothing but your payoff phase, because the energy is produced in the last 5 steps rather than consumed. It's very clear that in the first 5 steps, the energy will be utilized, and whereas in the second phase, we can see that energy is produced. Energy is nothing but in the form of the ATP molecules. The various enzymes involved in the next 5 steps are dehydrogenase, kinase, mutase, lyase, and kinases. First 5 steps of your glycolysis. Let us see one by one. As it's a glucose molecule, it starts with a glucose molecule. Glucose is nothing but a 6 molecule, 6 carbon moiety. As I told you, the enzymes involved are phosphorylation, isomerization, again phosphorylation, and followed by your aldolase. And the thing is, in this one, you can see that 2 molecules of ATP are utilized to undergo the further reaction. We can see in the first one, the glucose in presence of your ATP undergoes phosphorylation to form glucose 6-phosphate, and then your glucose 6-phosphate because of isomerization undergoes an isomerization reaction, and it is converted into your fructose 6-phosphate. The fructose 6-phosphate in presence of ATP undergoes phosphorylation, and then it forms your fructose 1,6 diphosphate. And the enzyme plays an important role by name aldolase. Aldolase in which your 6 carbon moiety is converted into 3 plus 3 carbon moiety, that is nothing but dihydroxyacetone phosphate and glyceroldehyde 3 phosphate. This is the first 5 steps of the glycolysis, which is there, which is involved in the preparative phase. Preparative phase is nothing but something you're preparing. That's what when you're preparing it requires an energy. That energy is nothing but your source is nothing but your ATP. Here you can see clearly that 6 carbon molecule, the glucose, is converted into a 2 different molecule by 3 carbon atoms of 2 molecules. That is nothing but your glyceroldehyde 3 phosphate and dihydroxyacetone phosphate. The fructose, you can see the enzyme aldolase. Aldolase will convert your fructose 1,6 bisphosphate by the presence of an enzyme aldolase to glycerol dihydroxyacetone phosphate as well as glyceroldehyde 3 phosphate. In presence of an enzyme phosphate isomerase. Isomerase is something that it has the same molecular formula, but only it differs in the structural formula. You can see the structural formula difference alone in these 2 compounds. Let us go next to the next one. Then your glyceroldehyde, it enters into the pathway of payoff pathway. You can see here, you can know clearly that it enters one glucose molecule is split into 3 molecules, into 3 carbon moiety, to 2 molecules of 3 carbon moiety. So what will happen? Your glyceroldehyde 3 phosphate in presence of an enzyme, glyceroldehyde 3 phosphate dehydrogenase, it is converted into your 1,3 bisphosphoglycerate. In the bisphosphoglycerate, you can see that your NAD is converted into your NADH plus H plus ions, and your 1,3 bisphosphoglycerate is converted into your 3-phosphoglycerate in presence of an enzyme phosphoglycerate kinase, which will release 1 ATP molecule. The 3-phosphoglycerate in presence of your enzyme mutase, you can see the location of the carbon atoms in the third position. The phosphate group was there. After the enzyme mutase, the 3-phosphoglycerate will be converted into 2-phosphoglycerate. The phosphate group from the third position, it shifts to the second position. Then, in presence of your enolase enzyme, it plays an important role in the formation of your phosphoenol pyruvate. Then, finally, the kinase enzyme. The kinase enzyme is nothing but the pyruvate kinase. By the name kinase, we can see that there will be a release of an ATP. When you just take the word dehydrogenase, surely NADH plus H plus ions comes into an action, whereas in the kinase, ATP release will be there. So your phosphoenol pyruvate is known as PEP, that is converted into pyruvate. This is the steps, this is the last 5 steps which is involved in the glycolysis, which is also known as payoff phase. The payoff pathway. Reason is, in this one you can see that NADH plus H plus ion. When this NADH plus H plus ion enters into the oxidative phosphorylation reactions, then it enters into the electron transport chain. From the electron transport chain in the complex one, you can see that one, your NADH will release 3 molecules of your ATP. Remember, NADH will give rise to how many molecules? 3 molecules of ATP. So in the 6th step, you will get 3 molecules of ATP. Again in the 7th step, you can see that 1 molecule of ATP. So 3 plus 1, 4. Again in the next step, you can see that one more ATP is there in the 10th step. So 4 ATPs. So how many overall? Totally 3 plus 1 plus 1. Overall 5 ATPs are released during the 3 carbon atoms. That is only one glycerol-dehyde 3 phosphate can give rise to your 5 molecules of your ATP. Just imagine glyceraldehyde along with dihydroxyacetone phosphate. So you have to multiply into 2. So 5 into 2, overall 10 molecules of ATP is released during the glycolysis formation. As I told you, the preparative phase, how many ATPs has been consumed? That is nothing but 2 molecules of ATP has been consumed. So this is the overall schematic representation of your glycolysis. You can see from the glucose till pyruvate. This is the aerobic respiration, aerobic glycolysis, what we are discussing now. From the glucose to the pyruvate, you can see that 2 molecules of ATP are utilized in the first and the third step. After that remaining, you can see the 6th step, and then the formation of your ATP will be there. So when you multiply with 2, it's overall 10 molecules of ATP has been released during the payoff phase, whereas the preparative phase, 2 molecule of ATP has been utilized. So 10 minus 2, it is an 8. So overall 8 molecules of your ATP is generated. This is the summary of your glycolysis. You can see that one, the glycolysis, which is converted into fructose 6-phosphate. This is the inhibitors. I can say an inhibitors. Each and every metabolic pathways, it has its own checkpoint as a, okay. The inhibitors in the glycolysis. In this summary, I have shown you both the glycolysis as the gluconeogenesis. The reason behind this is the glycolysis is the pathway in which you can convert the glucose to pyruvate, whereas your gluconeogenesis is the pathway in which your pyruvate is converted into your glucose. The only 2 different enzymes are involved in that one. One is your pyruvate carboxylase and one is your phosphoenol pyruvate carboxykinase. This is the reverse of your glycolysis. It's very simple to remember that glycolysis, the reverse of your glycolysis is gluconeogenesis. The only 2 different enzymes involved here are pyruvate carboxylase and the phosphoenol pyruvate carboxykinase. The inhibitors in your glycolysis is nothing but your ATP, citrate, and H plus ion, which will inhibit the formation of your fructose 6-phosphate to fructose 1,6 bisphosphate, whereas your phosphoenol pyruvate to pyruvate is inhibited by your ATP and alanine. And in case of your enhancement, then you can see that the fructose 1,6 bisphosphate will be as a stimulators for the formation of a glycolysis. At the same time, you can remember that the inhibitors ATP, citrate, and H plus ion, ATP and alanine in case of your glycolysis. The energetics, I told you already that in the preparative phase, for the first phase, the first 5 steps, there will be utilization of ATP. That's what we are reducing, minus 2 ATPs. Whereas in the next step, you can see that 3 as 1 molecule of NADH, as we are undergoing 2 steps, so 3 ATP plus into 2 will give rise to 6 ATP. In the step 6 and 9, you can see that 2 ATP into 2 molecules will give rise to 4 ATP. Overall, in the aerobic glycolysis, 10 molecules of ATP will be released minus 2 molecules of AATP is equal to 8 ATP molecules plus glucose YT. So overall, during the glycolysis, from the conversion from the glucose to the pyruvate, totally 8 molecules of ATP is released. Whereas in the glycolysis, you can see in the 1 and 3 step, the same 2 molecules of ATP will be used. Whereas in the 6th step, 6th and 9, 2 ATPs will be released. and when it is occurring twice the time, so into 2, so 4 minus 2, overall 2 ATP only is formed during the anaerobic glycolysis. But the aerobic glycolysis, the end product is pyruvate, whereas in the anaerobic glycolysis, the end product is what? Lactic acid. Let us see the clinical significance of the glycolysis. Each and every metabolism, there is some clinical significance are associated. The pyruvate kinase deficiency is an autorecessive mutation that will lead to an anemia, which is nothing but hemolytic anemia. In this hemolytic anemia, what happens exactly is there will be an inability to form the ATP, which because of this inability that will causes your cell damage, and because of cell damage, what will happen? The cells will become swollen and it will be taken out by the spleen, which will lead to a splenomegaly. That is nothing but your inflammation in your spleen. The mostly inflammation is with that symptoms will be your jaundice, icterus, and elevated bilirubin. As we know that something it's related to bilirubin is nothing but your related to your spleen or as well as your liver, and then splenomegaly. As I already told you, the energetics of the glycolysis. This is to once again to recall. I will elaborately given the glycolysis is nothing but 2 molecules of NADH will produce 6 molecules of ATP. 4 ATP will produce 4 molecules of ATP, and 2 molecules already which is used in the preparative phase. So overall energetics is 8 molecules of ATP per molecule of glucose for 2 molecules of your pyruvate formation. In the right corner, you can see that the oxidation of your NADH will always release 3 molecules of ATP, FADH will release 2 molecules of ATP, and GTP will release 1 molecules of ATP. Thank you. Let us see in the next video the continuation of your gluconeogenesis in future.